que tiene un problema en mi laptop y puede iniciar, es poco tarde para iniciar. Y por eso yo espero para solo, por favor puede esperar unos dos, yo no conozco, pero yo checo y yo puedo iniciar mi presentación. Solo, por favor, es poco tarde en mi laptop, ¿ok? Por favor, una gran disculpa para molestia. Sí, que es... oh, hola, buenas tardes. Perdón, ahorita, ¿se puede escuchar bien? Perdón, no escucho. Ah, sí, perdón, ahorita, sí, ahorita yo puedo escuchar. Perdón, ok. Iniciamos de presentación, pero primera cosa es que, por ejemplo, usted tiene algunas dudas, algunas preguntas, cualquier cosa usted tiene, sí. Por ejemplo, usted tiene alguna duda, usted puede preguntar, ¿ok? Sí. Usted entendí todo de conceptos desde 2 días, ayer y también de órgano on a chip. Y cualquier duda usted tiene, por favor, puede preguntar, ¿ok? En cualquier tiempo hoy o en la tarde. Y yo también puede mandar presentaciones para usted, ¿ok? No tiene ningún problema, ¿ok? Sí, ahorita abriendo. Ustedes trabajando en sensores y en parte de sensores hay muchas cosas que es muy crítico en sensores, ¿no? Más importante es que hay diferentes tipos de sensores. Hay óptica, hay también electroquímica, ¿sí? Eso es, hay muchas diferentes tipos de, de cosas. Y ahorita iniciamos con quimosensores. Mm, Quimosensor. Perdón, perdón. Esa es ok. Mm. I'm sharing my screen here, and here we have. This is a presentation of one of my professors who is actually working on chemosensors, and he was kind enough to provide me with a complete presentation because he used to give us a lecture, and I'm like continuing the same the thing, and it was really great honor. It was a great honor to work with him. That was the thing, and that's the reason we have been. It's a huge presentation. I will not be taking this presentation in details. Like it's not necessary also for your curriculum. But then this particular when you talk about chemosensor, there are very important aspects whenever you're talking about chemosensing. So a chemosensor, what happens is there is a molecule which will bind. So there is a molecule which will have two active sites. It will have two active sites. One active site is wherein the molecule will bind to the ligand. Ligand, I hope you all understand ligand. So there is one active site which will bind to the ligand, okay? And this active site is considered to be also known as receptor. And this receptor and ligand interaction happens. So once a receptor ligand interaction happens, this complex will then give signals in the whole molecule. This complex will give signal, okay, to the fluorescent compound which is attached to the same structure. For example, this is the molecule you can see here. This is the molecule, and this particular substrate has got two active sites. On one active site, with the that is known as receptor, which will bind to this kind of ligand. And once the receptor ligand interaction happens, like this, once a receptor ligand interaction happens, then there is a conformational change. And when the conformational change happens, then this is illuminated. Then the whole complex gives illumination in terms of fluorescence. Without the ligand, there is no fluorescence. And with the ligand, immediately there is a conformational change that leads to fluorescence. I'll just show you, exemplify this with an example, with a video. I just want to share with you this. So here we have a chemosensor video. So I'll just show you how it works. But first, I would like to show you something about fluorescence and phosphorescence. Okay, I'll explain you that.
Just find a way to restore crystal clear 20/20 vision without. Explain the principle of fluorescence and phosphorescence.
I'll just share. I'm sharing it. Don't worry. Just give me a second. Here we have. So first of all, my friends, what you need to understand is what is fluorescence, and how and also what is phosphorescence. Because chemosensors, when you say, it deals with fluorescent compounds. So there is a chemical which is fluorescent in nature, and it is fluorescent under certain condition. That is not fluorescent throughout. It is under some certain condition. All right. And when this particular condition is fulfilled, then the molecule will exhibit fluorescence. Similar is the situation with phosphorescence also. But I would like to explain you here with a pictorial picture how it works, what are these compounds. Just I would like to give you an example here. Tell me if you can hear properly. Okay.
No.
Sorry, come again.
Que no se escucha.
No escucha. Okay, okay. Okay. Hmm. Okay, I will your strings enhanced encryption mode. Disable annotations, hide names, hide share sound.
Electronic energy level to high electronic energy level.
Can you hear now? ¿Puedes escuchar ahorita?
Sí, ahorita ya. Gracias.
This absorption of light requires 10 to the power minus 15 seconds. The instant re-emission of the absorbed energy is called fluorescence.
Okay, otra vez.
Many compounds absorb ultraviolet or visible light and undergoes electronic transitions from low electronic energy level to high electronic energy levels. This absorption of light requires 10 to the power minus 15 seconds. The instant re-emission of the absorbed energy is called fluorescence, while the delayed re-emission of the absorbed energy is called phosphorescence. For most molecules, the electrons are paired in the ground state, that is, pair of the
Okay, I have to
absorbed
explain.
energy is called fluorescence, while the delayed re-emission of the absorbed energy is called 15 seconds. The instant re-emission of the absorbed energy
I have
is
to.
called fluorescence, while the delayed re-emission of the absorbed energy is called
Ahorita, usted quiere entender esta información, ¿no? Cada vez mi hija usó Annotate, Remote Control, AI, Summary. ¿Ustedes conocen dónde para escribir cosas aquí, perdón?
Hay como una flechita arriba, creo que le aparece como verde y negro en la parte de hasta arriba de la pantalla.
Verde y...
O donde está lo de su micrófono y todo eso, hay como una flechita. No sé si le aparece.
Y en verde tiene en Select Your Speaking Language, you're using Enhanced Recording to the Cloud. Todo está bien. Pero aquí hay apps. This Remote Control, Annotate. Es Annotate. Ajá, eso. Pero yo quiero una blanca para escribir, como pizarrón, ¿no? Así. Perdón, otra vez. Yes, whiteboard, ya. Ok. Apps, ok. Apps, un segundito. ¿Dónde es? Ok, así. Ahorita no puedo, ok. Perdón. Ok. Apps. Virtual backgrounds? No. Oh, God. Oh. Un segundito, yo pregunto con mi hija. Un segundito. ¿Aquí? Oh, whiteboard, ¿qué es el canto? Sí. Ella es mi hija. Ella conoce más. Ay, espérame, un segundito. Sí, ahorita. Ok. Whiteboard. Anteriormente no puedo ver. Y bueno, muchas gracias, señorita. Ok. ¿Dónde voy a escribir? Hay plumas. Ok. More board references. I think हो जाएगा मैं कर लूँगा. Okay. Okay. लिखने को नहीं हो रहा है. Okay, no problem. I'll manage. Okay. Okay, I'll manage. Don't worry. It's done. Same organization. Ah, okay. Nosotros no puede hacer. We cannot do this. Está bien. Okay. दरवाजा बंद कर लीजिए सर. Can you close the door? Right. Goodbye. Yeah, this is the pencil, right? Yes, perfect. Thank you. Yeah, here. That. Okay. Share screen. This one. Perfect. Thank you. Gracias. Trata con perdón. Okay. Anita, ¿sí puede escuchar todos? ¿Está aquí o...?
Sí, sí se escucha. Sí está.
Sí.
Gracias. El problema es que no va... puede abrir whiteboard en Zoom, pero nosotros no tenemos, pienso que, permisos. El mismo institución sí tiene permisos, pero afuera de institución no tiene permisos, dar permisos por eso. Eso escribió, pero no hay problema. Mi hija tiene diferente idea, por eso ahorita mando este pencil, ¿ok? So, what is fluorescence? Now try to understand what is fluorescence. Anybody knows what is fluorescence, first of all? I should have asked you first, guys. What do you mean by fluorescence? Sorry.
Fluorescence, it's when, sorry, go ahead.
Yeah, tell me.
We were discussing about the difference between fluorescence and chemioscence. For example,
What fluorescence?
fluorescence, it's when a compound, let's call in a formal way like that, or an object or whatever, keeps on its well, it keeps electrons, in this case photons, well, or energy, which is then expelled in a way of photons, so that so in that way we could see light. Is this something kind of related?
Yeah, but I can explain in a pictorial way. Fine, it's okay. But I'll just show you here. Okay. There is a electron which is moving from, this is the ground state, and this is the excited state. Okay? And the electron is here. This electron moves from the ground state to the excited state when light or a photon is hitting, like I mean to say there is a photon here. And when the photon is hitting this electron will move up and it will sit onto the top. So once it sits onto the top, it is known as excited. So this is excited state. Okay. And this is the ground state. So when the electron is moving on the top, then it will be staying there. It will be stalled over there for 10 raised to minus 15 seconds. So it will be stalled over there for 10 raised to minus 15 seconds. And what will happen after that is the electron, this electron will come back onto the ground state again. It will come back to the ground state after 10 raised to minus 15 seconds. So once it comes back to to the ground state, it will release some wavelength of light. Now, guys, I want to ask you one question. Here, there was one particular wavelength. And then this excites the electron. Electron stays over there. And then electron comes back to its ground zero state. And then there is photon which is being released. So now this photon will have higher energy or lower energy in comparison to this light. Or you can say, what will be the wavelength? The wavelength of this particular light which is emitted will be higher than this or lower than this. It's a question for you guys. Should I share it with you again? Don't write in the chat because I will not be able to see the chat. So please don't write in the chat. I can see the chat also, I think. No, I cannot see the chat. Yeah, I can see the chat. Yeah. Can you, guys, could you answer me this question? Maybe who wants to answer? Enrique? He says is always like he has to answer always questions. So I would like to ask Enrique and CISO. All right, Francisco Josue. No. All right, all right. I hope you are you know what is the like how to calculate the wavelength. What is the relationship between wavelength and energy? I think you all are aware of that, right? E is equal to h nu. Do you know that? Or E is equal to, okay, E is equal to h c by lambda. This is a wavelength. All right, I will explain completely. No worries, I understood that. All right, fine. The thing is, there is a wavelength, there is a particular wavelength of light here, which is passing. And there is a particular energy of the light which is passing through this. And that particular energy of light will activate the electron. Once the electron is getting activated, then after that, you can find once the electron gets activated, it will be stored here. All right. So what will happen there when the electron takes up the energy, the photonic energy, it takes up the photonic energy, it stays on the top, it goes on the top, and it stays over there. And during that process, it loses little bit amount of energy. Okay. And then when it's losing the energy, okay, it stays there for 10 raised to minus 15 seconds, it loses, it radiates out energy, it gives out energy. All right. Moreover, there are chances that there can be some sub levels or some energy levels, etc., etc. But anyways, in this case, I consider that there is not much energy levels. There are no much energy levels, like, because so here what will happen is in fluorescence, the electron is excited because of the photonic energy. It will stay here and it will lose some amount of energy while doing this job, and then it will come back to the ground state. And when it comes back to the ground state, it has lost some amount of energy from the previous photonic energy which has been taken here. So it has lost some amount of energy from this photonic energy, from this photon, and now this particular electron will release that energy. So what do you think? The energy, as I told you, energy is decreased, right? So initially the energy was higher, but when it releases, like when it comes back, the energy is lower. And you know the relationship. When energy and wavelength, they are inversely proportional to each other. So when the energy is lower, the wavelength is higher. So for example, here I have a wavelength of light, which is approximately 400 nanometer. This much of energy, this particular light is being absorbed by the electron. And then electron gets excited. And this same, after losing some amount of energy, it comes back and it releases the energy, it releases the photon back. So this time, can anybody tell me what will, like, it will be more than 400 or it will be less than 400? Oy.
More?
Again, doctor, tengo problemas en la conexión. Ok, ok. Yo explico otra vez. Enrique, perdón, yo no puedo ver de este mensajita, una gran disculpa. Porque cuando nosotros puede compartir, de todas las cosas está desagregada, ¿no? So, ahorita yo puso de chat también está abierta, ¿ok? No hay problema. Pero una gran disculpa que yo no veo de este mensaje. Ok. My question again. There is a wavelength, this particular wavelength of light, which has activated the electron. Electron gets excited, comes back to the ground state after some time, and then it releases the photon. Now, what is the wavelength of this particular photon? Is it more than 400 or is it less than 400?
More?
You're saying more?
Yes.
Okay, somebody else?
I think less.
Sorry?
I think less. Cause
Aranza thinks less.
yeah,
Okay.
cause they lost energy.
Just want to check. You can put thumbs up if you think that it will be more. And I think there is no thumbs down, it seems. So you can just simply don't, if you think it's going down less than 400, then you don't need to do thumbs up. How many people think that it will be more than 400? Oh, okay. Miriam, now you don't want to say, okay. Miriam, Sergio, Jose Valentin, Enrique. Okay. All right. Others are saying it will be less than 400 or they don't have the answers, right? So the answer is it will be more than 400. Why? Why it will be more than 400? Anybody can
Por
answer
la
that?
ecuación,
Perdón.
por la ecuación. O bueno,
Por
es
la
que
ecuación,
dijo
correcto,
que
pero
si la energía es
lógicamente.
menor, entonces quiere decir que, pues, por la ecuación la longitud debería ser mayor.
Correcto, correcto. So when there is increment, for example, sorry, when there is like when the energy is decreased in comparison to the initial energy, when the energy levels go down, then after that, what happens is the wavelength increases. So maybe the wavelength can be around 500 nanometer or 450 nanometer, I should say. It's not that big, but yeah, 450, 480, that will be the difference. All right. So this is what is happening when you are having, what do you call? There's no rubber. Yeah. So this is what is happening when you are having, yeah. Yeah. Okay. For fluorescence, okay. But let's talk about phosphorescence. Again, same line. This is ground state. This is excited state. This is the wavelength of light which is coming. There is a circle. This is the electron which gets excited. And the electron is here. Sorry, there is. Please don't consider that the size of the electron increases. Please don't consider that. It's actually the same size. By mistake, I have made this. Maybe I can remove this. Okay. So here we have the electron. Okay. And now once the electron reaches onto the top, it stalls here for 10 raised to minus 15 seconds. Okay. So you know that it stalls here for 10 raised to minus 15 seconds. Plus, now this particular electron doesn't come directly. It then moves to the second level, then moves to the third level, then moves to the fourth level. So it comes like this here, stepwise. It moves stepwise. And then finally, it comes here. And then it will show the electron. And now this electron is back to the position. But what will happen? And at this point, it will release the photon. So what will happen is it takes a lot amount of time. So there is a delayed emission. So when there is light exciting it, excitation happens, it stalls more in all the places and then returns back. It travels a lot and then it returns back. It loses a lot amount of energy and that leads to like the emission of light takes a lot amount of time. It's known as delayed emission. Okay, and moreover, it loses more amount of energy in this case. All right. So in the previous case, in fluorescence, it loses lesser amount of energy, but in this case, it loses more amount of energy. Could you understand this concept, guys?
Yes.
Perfect. Now, let's go back to the video. Oh my God, I lost the video. Okay. Yeah, it's here. Yeah. Okay. My God. Shit.
Phosphorescence. For most molecules, the electrons are paired in the ground state. That is, pair of electrons have opposite spin. This is called singlet state. As the spin is paired up, they do not show any magnetic moment. But if the electrons have the same spin, the magnetic moment of both the electrons gets combined and they behave like a tiny magnet. This state where the electron pair is having same spin are called as triplet state. In molecule, there are several electronic energy levels and electrons are present at the ground state in the singlet state, that is with opposite spin. When molecules absorb UV or visible light, one electron from ground state undergoes to an excited state. Now, excited state is also having several vibrational levels. So, the excited electron loses its energy by intermolecular collision and it comes to the lowest vibrational level of excited state. Finally, the electron from the lowest vibrational level of excited state return to ground state by emitting the radiation or light of lower energy or higher wavelength. This overall process of absorption, intermolecular collision and emission takes 10 to the power minus 4 to 10 to the power minus 8 seconds, which is very small. Thus, it looks like instant re-emission of light. But the energy of radiation emitted is always less than the energy of radiation absorbed. Or, in other words, wavelength of emitted radiation will be always higher than the wavelength of radiation absorbed. This is fluorescence phenomena. In case of phosphorescence, the inversion of electron spin takes place. That means when the energy from UV or visible light is absorbed, the electron from the ground state jumps to excited state. At the same time, the spin of the electron gets reversed and it is now in triplet state. That is, both electrons have same spin. In triplet state, there is less chance of electron coming back to ground state because pairing of electron with same spin is not favorable. Thus, it takes more time to reverse the spin and come back to ground state. The time taken by electron is from 10 to the power minus 4 to several seconds. Thus, it seems that the emitted radiation is delayed. In some cases, even when we turn off the light source, radiation keeps on coming. This delayed re-emission of radiation is called phosphorescence. Here again, the energy of radiation emitted is always less than the energy of radiation absorbed. Or in other words, wavelength of emitted radiation will be always higher than the wavelength of radiation absorbed.
He is my PhD colleague. This guy is my PhD colleague. So, yeah, he's a professor now in Italian Institute of Technology. Yeah, from Italy. Yeah, he's in Italy right now. Anyways, but then there is one more thing I would like to show you. Fluorescent biosensors. There are okay, first of all, there is one very important, yeah, here, friend. Now, guys, you will be taught what is the meaning of fluorescence resonance energy transfer. When we are talking about, okay, I'll just show you here. In this particular screen, if you can see this chemosensor, this particular presentation, what is happening is there is a receptor, this is a receptor and the ligand interaction. And then accordingly, there is a change. There is a conformational change. And then there will be some fluorescence, which will, like here, you can see there is some fluorescence seen. All right. So there is there is some energy transfer happening, and then that leads to the fluorescence. But moreover, there are certain things like infrared, there is a little different, wherein initially, like there are two fluorescent molecules which are all like, for example, okay, you can see here. In this case, this is one active site and this is another active site, for example. And as soon as this ball interacts with one active site, then this complex, there is a conformational change and both the active sites will come together. And once they come together, then there is fluorescence or loss of, or it can be anything. Either there will be fluorescence or there will be loss of fluorescence. Anything can happen. And if they come back, it is dependent on the behavior of the molecule, right? So, and if the molecule is in a straight form, then they will be non-fluorescent. If they come like that, then they become fluorescent. And what they do is they transfer the energy also from one fluorescent molecule to the other. So this is what is known as fluorescence and resonance energy transfer. So I'll just but you can understand it pictorially with this particular presentation. It is like really a good concept. So I would like to teach you this also. Please listen to this. I would like to, oh, sorry. Yeah.
Back in third quarter planning, Adams is quarterbacking this project and
Yeah.
Hey everyone, quick back to Mr. Basics here. Let's talk about fluorescence resonant energy transfer, or FRET. As the name suggests, fluorescence occurs when there is a resonant energy transfer between the two molecules. The molecule that accepts the resonant energy is called the acceptor, while the one that gives the resonance energy is called the donor. Let's understand this with an example. Let's assume that we have two proteins, CFP and YFP. CFP is the donor, while YFP is an acceptor. When CFP receives energy from the excitation...
Okay, I can show you something else. This is single molecule FRET.
At St. Jude Children's Research Hospital, we are dedicated to developing new technologies to investigate serious diseases. Cells, the building blocks of our body, contain trillions of unique biomolecules and complex molecular assemblies that maintain the cellular functions important to human health. By investigating the molecules and assemblies responsible for these functions, such as machinery that degrades proteins, and ribosomes, which are responsible for making new proteins, researchers can better understand the molecular basis of disease and develop effective therapies. Conventional techniques to visualize cellular components typically probe billions of molecules simultaneously, which often obscure the behavior of individual molecules. In the Single Molecule Imaging Center at St. Jude, we engineer and employ extremely bright fluorescent probes in order to investigate individual molecules and their interactions. To obtain accurate images, we use highly advanced custom-built microscopes. Multiple lasers are co-aligned and then focused by a series of high-power lenses and other specialized optics to a very small volume. The focused laser beams illuminate the fluorescently labeled molecules, and the probes emit light in a manner that reports on their positions and movements. When the probes are close together, more red light is emitted. When the probes are far apart, more green light is emitted. This approach, called single molecule FRET imaging, allows us to...
Could you understand this? Hello. Hello, I could not hear. Could you understand?
Yes.
Okay, so you could understand fluorescence, resonance, energy transfer, and you could understand that when they are away, they will give different fluorescence. And when they are nearby, they will give different fluorescence. But you understand? And how so this particular video which you have seen, it is actually telling you the function, what is the exact function. So whenever there is a ribosomal RNA, ribosomal system, and there is a protein synthesis happening, so if we want to understand that actually where this, in which particular site, where is exactly the protein synthesis happens, okay, if you want to understand the mechanism, then you can just have labeling of two different zones, okay, and around the active site. And whenever there is some protein getting in, then what will happen as soon as the protein gets in, it the two ends of the active site will come together. And if the protein comes out, it will go back. Protein gets in like this, like ribosomal RNA and the mRNA. mRNA gets in, it will be it will be like this. mRNA gets out, it will be like this. So in this way, you can understand that how what is how many seconds it takes for a particular protein to be synthesized, or microRNA is synthesizing, is connecting all the amino acids together, right? Like that. tRNA, mRNA, and ribosomal RNA, how all of them collate, connect each other, and then allow the amino acids to get connected with them. So this is a beautiful presentation. I would like to again restart. Will that be okay? You want to see it again?
Yes, please.
Okay, I'll show you again. This one is really very interesting. And it's a live example. I'm going to say like pictorial, but of course, but it's a live. So you can see
Technologies to investigate serious diseases. At St. Jude Children's Research Hospital, we are dedicated to developing new technologies to investigate serious diseases. Cells, the building blocks of our body, contain trillions of unique biomolecules and complex molecular assemblies that maintain the cellular functions important to human health. By investigating the molecules and assemblies responsible for these functions, such as machinery that degrades proteins, and ribosomes, which are responsible for making new proteins, researchers can better understand the molecular basis of disease and develop effective therapies. Conventional techniques to visualize cellular components typically probe billions of molecules simultaneously, which often obscure the behavior of individual molecules. In the Single Molecule Imaging Center at St. Jude, we engineer and employ extremely bright fluorescent probes in order to investigate individual molecules and their interactions. To obtain accurate images, we use highly advanced custom-built microscopes. Multiple lasers are co-aligned and then focused by a series of high-power lenses and other specialized optics to a very small volume. The focused laser beams illuminate the fluorescently labeled molecules, and the probes emit light in a manner that reports on their positions and movements. When the probes are close together, more red light is emitted. When the probes are far apart, more green light is emitted. This approach, called single-molecule FRET imaging, allows us to track the movements of each part of the single functioning molecule in real time. Characteristic features of the FRET signals originating from individual molecules can reveal both how they correctly function and how they malfunction. These discoveries could lead to new treatment options and more effective clinical care for patients.
So could you understand, guys? So here we have been dealing with how the fluorescence energy transfer takes place. So and similarly here also there is like I'll just share with you the screen. Here also is like energy transfer which is happening, and then that leads to fluorescence of the molecule. So you can see here So here you can see it is a chemosensor that generate a fluorescence signal. So now you understood. So what is a chemosensor? Chemosensor, it can be used for understanding luminescence. Okay, it can be like you can exploit whenever you say chemosensor, it can exploit either fluorescent molecules, or there can be absorbance, there can be redox potential change, there is a redox change, or there can be an NMR relaxation types, like using NMR sensors, um, nuclear magnetic resonance imaging or nuclear magnetic resonance sensing. You can identify what is the how the molecule looks like, and you can identify the molecules for sure. So fluorescence chemosensor means what? You know, it is a big issue that this particular sensor is basically when we talk about fluorescent signal, that means there are different types of fluorescent molecules like TRITC. TRITC is tetrarhodamine isothiocyanate. There is another molecule which is known as FITC, which is known as fluorescence isothiocyanate. All right. So these are the different kinds of molecules. These are two different kinds of, there are many, cyanin, cyan red, or there can be many different kinds of molecules which are fluorescent in nature. And then doxorubicin is also a kind of a drug, which is also fluorescent in nature, which can be used for detection. Like based on the fluorescence, if the molecule is exhibiting this fluorescence state, then using this, what do you call, using these sensing systems, using bioreader, using this fluorescent reader, you can detect the molecule. Okay, you can detect whatever molecules you want to detect using this fluorescent sensing system. So why fluorescence? Because it's highly sensitive, highly spatial and temporal resolution. Spatial is space and temporal is time. So it is rapid. So it takes just 10 raised to minus 4 or 10 raised to minus 8 seconds to receive the signals. And then it is cheaper. Okay, it's easy to perform. That's the idea. And you have already, so you know what is photoluminescence or fluorescence. So what happens is there is an excitation of the electron from one particular energy level. It reaches to like another energy level, and then it will come back to its ground state. So this is the excited state, and this is the ground state. So when it comes back to the ground state, there is emission. So this is also known as fluorescence. All right. And how can you measure the signals, the fluorescent chemosensors? How can you make it? First, by quenching, fluorescence quenching. That means when like if at all, like here you can see here, like this molecule sits here, then there is fluorescence. If this molecule is gone off, there is no fluorescence. So you can design a sensor based on this particular complex. So if this complex is removed, there will be quenching. There is no fluorescence. And if this molecule comes, there will be fluorescence. So this is the idea. Okay. So what we can measure, one is fluorescence quenching or fluorescence increase, or you can measure emission spectrum. If there is a shape modification, that means the intensities are higher or lower, you can find out that. You can find out the lifetime, that means how much time it takes for the fluorescence to stay, maybe 10 raised to minus 4, 10 raised to minus 8, or something more or less. Emission and isotropy. So there will be emission of the molecule, and how the emission takes place, that critical understanding you can have when you're dealing with emission and isotropy. So then there are, I think it's the same. Oh, sorry. So here you can see that there is a strategy wherein there is a receptor, the ligand will interact. When there is a receptor, there will be fluorescence. And when the ligand sits onto it, there is no fluorescence. So this is one type of fluorescent molecule. There is another type of fluorescent molecule wherein this particular complex, there is a complex formation. So this fluorescent probe will be interacting with the molecule, with whatever structure or a compound. And when there is nothing onto the top, there is a receptor is empty, then this particular molecule which is associated with it, it will fluorescence. It will show fluorescence. And if as soon as the ligand sits onto it, there is no fluorescence. Here, like a competition, okay, there will be competition. So when there is receptor ligand interaction, normally there will be fluorescence. But if there is there is an analyte coming from the sample, it comes and occupies this seat and replaces this particular fluorescent molecule, then after that you will find that the fluorescence has lost. So this is the mechanism. So individually also you can see here the ball, this particular ball, whenever it is reacting with the receptor, then it will show fluorescence. But as soon as this molecule this ligand is out, it will not show any fluorescence. All right. Okay. So now, so there are different molecules which show fluorescence. One of them is Fura2, Keen2, Mag-Indo1, CN also. So what happens is when calcium, so it is dependent on calcium. So it detects calcium ions. This guy detects magnesium ions. This guy detects sodium ions. It's like that. Guys, you want to take a break for 5 minutes and then we can join again. Okay?
Yeah, thank you.
Thank you. Can we start again, students? Hello?
Hello.
Ya, gracias, gracias.
Yes,
Otra vez, iniciamos con respecto de, perdón, de, I'm sharing the screen. Okay, okay, here. So you can see here, there are different kinds of sensors. This is a calcium sensor. For calcium sensor, you are having FURA2. Then there is magnesium sensor. You can have MAG-INDO1. And for sodium sensors, you can have CN. These are the three different kinds of sensors. Now, do you know where these calcium sensors are being used? For example, if you want to detect the calcium signaling pathway, all right, there you will be using this kind of calcium sensor. All right, it will be a live sensor. And for example, in the neuronal signaling pathway, there is a major role played by calcium. So calcium is a very important molecule which needs to be detected so that we can understand what are the effects of calcium signaling, how the calcium signaling pathway occurs. So you want to understand that, I will again give you a pictorial picture, like why calcium signaling, fluorescence-based calcium signaling is very important, and how is it useful, how critical it is. You can understand uh, okay.
From repulsive odorants. Numerous biological phenomena can only be witnessed at the subcellular level. For example, choreographed chromosomal couplings and uncouplings at cell division, the morphology of circulating red blood cells, and neuronal activity occur at a resolution below human vision. Given that the resolution of the human eye.
So you can see here, there are neuronal signaling pathways wherein one neuron will pass on the potential, like through the potential generated through this, it can pass on the signals. All right. So maybe neuronal signals. So this is the way how neuronal signals move. That means these are sheet, and these sheet are allowing the signals to move rapidly, and there is like too much of calcium during this process. There is too much of calcium, like polarization and depolarization happening, and that polarization or depolarization which happens in the neuron is because of the calcium signaling, and even sodium potassium signaling, like that. I'll just show you how it works.
Is insufficient to distinguish or even perceive phenomena at the subcellular level, how are scientists able to visualize cellular and molecular interactions? Modern recombinant DNA technologies and microscopy can be combined to modulate and visualize molecular interactions. As an example, fluorescent calcium imaging can be used not only to observe neuronal.
So you can see here that where is the calcium present. So when you add a fluorescent dye, which can absorb calcium, which can interact with calcium, and as soon as it enters, so this is the fluorescent dye, and I attach calcium in this. Okay, calcium gets into it, then this particular molecule becomes fluorescent. So this is the greatest ever example. So this is your this is the neuronal cell having multiple sheets into it. All right, and then in all of them, how much concentration of calcium exists. So you can see here, there is a very high concentration of calcium is present during the polarization depolarization process. So here itself, you can understand that calcium plays an important role in transmission of neuronal signals. If calcium doesn't exist, there will not be any transmission of the signals. So here itself, it's a greatest ever example that calcium plays an important role in neuronal signaling. Yes.
Activity, but also to associate behavioral characterizations with physiological states. In our research, we use calcium imaging via recombinant DNA technology and microscopy to not only observe neuronal activity, but also to associate behavioral characterizations with physiological states. In order to detect the movement of calcium ions during neuronal signaling, a genetically engineered calcium indicator, or GCAMP, is required. GCAMP is a hybrid calcium sensor encoded by the calcium-binding messenger protein calmodulin, the calcium N-calmodulin binding M13 peptide of myosin kinase, and the fluorescent protein GFP. Few interactions are maintained between the GCAMP protein domains, and as a result, GFP fluoresces minimally in the absence of calcium. In the presence of calcium, calmodulin undergoes a conformational change and is free to bind the M13 domain. This induces GFP to adopt optimal conformational changes for increased fluorescence. The increased fluorescence can then be detected visually under a microscope.
You can see here,
Calmodulin undergoes a conformational.
please try to understand this.
Few interactions are maintained between the
when
GCAMP
there is
protein
no
domains, and as a
calcium,
result, GFP
you can see
fluoresces.
here that this is a calmodulin complex. This is a calmodulin protein, which is actually present inside the cell. This particular protein will be interacting with calcium normally, and it forms a calcium calmodulin complex. But if I want to understand that there is a calcium binding, or there is no calcium binding, this is the best example that protein, the calmodulin, will be interacting with GFP, green fluorescent protein. And as soon as, if there is no calcium interacting, then this particular calmodulin protein will interact with green fluorescent protein, and there is no fluorescence seen. But when
minimally in the absence of calcium. In the presence of calcium.
Yeah. Now, in the presence of the calcium, you can see the calmodulin complex is open, and now due to the presence of the calcium, there is a change in the conformation of the whole complex. Previously, you can see here, it's such a small molecule, calmodulin. It's like very complex, compact, like this. But as soon as calcium comes up, it becomes a little bit inflated. It will become like this, you can see here.
In the presence of calcium, calmodulin undergoes a conformational change and is free to bind the M13 domain.
And then leads to,
this induces GFP to adopt optimal conformational changes for increased
as soon as GFP comes nearby, then this becomes highly fluorescent.
Fluorescence. The increased fluorescence can then be detected visually under a microscope. Let's see how we can visualize these molecular changes at the cellular level. We can inject model organisms such as the nematode C. elegans with plasmids that contain genetic sequences that will give rise to a calcium sensor protein. As a result, we can observe the relative changes in calcium levels in the cells of an organism over time. Calcium signaling plays an essential role in neuronal signal transduction. Let's look at C. elegans as an example. Calcium signaling.
So now here, in this particular concept, there is calcium imaging in Senorrhabditis elegans. Actually, there was a Nobel Prize winner who was working on Senorrhabditis. It's known as Senorrhabditis elegans. I can write it on your chat. I am such a dumbo. Wait a minute. Okay. So, like this. Elegans. Senorrhabditis elegans. This is the name of the worm, and this has got great amount of calcium in it, too much amount of calcium. And it's a very great model organism for understanding calcium signaling pathways. And through this, we could understand lot amount of signaling mechanisms inside the neuronal cells. So because since we could take the Sinorhabditis elegans and under the microscope and with all the fluorescent microscope, we could understand how how these calcium molecules can be trapped, and then we could understand how the fluorescence can take place, and that's the idea. And when this Sinorhabditis is like multiplying, how cell division happens, how this calcium is moving, and how and in what particular process. For example, calcium has got a great role during division. Calcium has great role during transmission of the signals. All right, magnesium has great role in during the replication, DNA replication. Magnesium has got a great role like that. So you can, how could you find that magnesium has got a great role? Can anybody tell me? How could you find that magnesium has got a great role in DNA replication? Here, calcium has got a great role during neuronal signaling, neuronal signaling pathway. Then sodium has got another role during Na plus K plus pump in mitochondria. So how could you find out that these particular ions are playing an important role? How could you find out? Can you tell me, guys? All right. So all these pathways happen. Oh, sorry, I could not. Okay.
Elegans as an example.
Here you can see the
Calcium
pathways.
signaling is crucial for C. elegans chemosensation. Previous research has shown that these microscopic animals can detect and move toward attractive odorants and away from repulsive odorants. When a worm is exposed to odorants, we can observe synaptic inputs and neuronal firing by measuring the relative changes in calcium levels in each neuron. Calcium imaging allows us to observe neuronal activity and associate behavioral characterizations with physiological states. C. elegans have 302 neurons, many of which are important for how the animal senses and reacts to stimuli. The AWC sensory neurons are located in the head of C. elegans and are critical for food-seeking behavior and chemotaxis toward volatile odorants. Behavioral assays suggest that the AWC sensory neuron responds specifically to attractive odors. There are two AWC neurons, the off neuron and the on neuron. Calcium imaging allows us to look at a neuron's activity as it happens. While a worm is immobilized, a specially designed microfluidic chip exposes the worm's nose to a flowing stream of test solution. Typically, when a neuron is activated, the concentration of calcium increases inside the neuron, causing the calcium sensor to activate. The fluorescence microscope allows us to observe fluctuations in neuronal activity. Now let's flow in our two stimuli. One stimulus, isoamyl alcohol, is an attractive odor, while the other stimulus is a neutral odor. Surprisingly, we see a decrease in intracellular activity when we flow in the attractive stimulus, and an increase in activity when we remove the odor. As it turns out, the active AWC neuron is an odor-off neuron that is activated by odor removal and inhibited by the continued presence of odors. This data, along with the previous behavioral experiments, has allowed us to determine that AWC responds specifically to attractive stimuli. Calcium imaging allows us to correlate behaviors with physiological responses in neurons. It is an important tool for providing highly quantitative data on neuronal activity.
So you can see here,
Intracellular
when the Sinorhabditis elegans are moving towards particular region, which is got attracted towards odorant molecules, then there is too much amount of calcium secreted, and then that leads to, this is completely fluorescent. All right.
calcium signaling has also been implicated in a host of human diseases, such as schizophrenia, Alzheimer's disease, and Huntington's disease. Visualizing the movement of calcium ions within a neuron is important in order to understand the pathophysiology of these diseases.
Okay. So guys, you have understood this, I hope. Hello. Hello, can you understand? Could you understand this? Well, if you have any doubt, please ask me if there is any question.
Profe, ¿será que podría repetir la segunda parte de nuevo? Es que justo se me trabó, entonces no pude entenderlo tan bien. La segunda parte del video.
¿Usted quiere de video o usted quiere que yo pueda explicar?
Pues si puede explicarlo, también estaría bien. O sea, como para entenderlo, es que no pude entenderlo.
No, está bien, está bien. Ok, no hay
Gracias.
problema. Sí. De cosa es que usted puede ver aquí. Okay. Now this is Caenorhabditis elegans. Okay. Now this Caenorhabditis elegans will be attracted towards the odorant. And as it is getting attracted towards odorant, what happens within them inside the Caenorhabditis elegans is there is a good amount of calcium signaling pathway, calcium signaling which will be activated. And these, because of the very high calcium signaling, they will be motivated to move towards the odorant, attractive odorant. So they will be like motivated to move.
When a worm is exposed to odorants, we can observe synaptic inputs and neuronal firing by measuring the relative changes in calcium levels in each neuron. Calcium imaging allows us to observe neuronal activity and associate behavioral characterizations with physiological states. C. elegans have 302 neurons, many of which are important for how the animal senses and reacts to stimuli. The AWC sensory neurons are located.
So these are the points that you can see they are looking like a neuronal cells only. So there will be a signaling pathway wherein, this whole area will be responsible to have calcium. So in a similar fashion here also, there will be too much of calcium and accordingly, they will be looking fluorescent. And moreover, this fluorescence will not be for a long time. There is one, for example, there will be one confirmation, like if they want to send the signals, there will be one confirmation. If they don't want to send the signals, there will be another confirmation. If they want to send the signals, there is too much of calcium secretion, and then your fluorescent molecule will detect the calcium. And then the whole thing will be fluorescent. If they don't want to send the signals, then though there is no calcium secretion, then there is no molecule which will bind calcium and become fluorescent. So that then it whole thing will be non-fluorescent.
In the head of C. elegans and are critical for food-seeking behavior and chemotaxis toward volatile odorants. Behavioral assays suggest that the AWC sensory neuron responds specifically to attractive odors. There are two AWC neurons, the off neuron and the on neuron. Calcium imaging allows us to look at a neuron's activity as it happens. While a worm is immobilized, a specially designed microfluidic chip exposes the worm's nose to a flowing stream of test solution. Typically, when a neuron is activated, the concentration of calcium increases inside the neuron, causing the calcium sensor to activate.
I think this is not important for you guys now, but this is the way your fluorescence activity is detected. Then, so what happens is, what you can see here, the applications like FURA2, it will be detecting calcium. And you can see here that you will be labeling it with the cells. And once the cells are being labeled properly, then you can find the color. These are the colored images and these are the black and white images. And you will understand how these molecules play an important role. Now, next. This is excitation intensity. So here, this FURA2, if I want to find out what is, how is it that these guys will be playing an important role. The best option is they will be they will be excited at different wavelengths. All right, this FURA2 will be excited at different wavelengths. So they will, as soon as they excite, at the concentration is increasing, the excitation of the molecule increases, and then finally a stage will come then where they will emit the radiation. Emission will be happening at lower radiation. So now, that there is internal energy transfer. So what is happening here is that this is the structure, this is the molecule of the, this is the molecule. I'm going to say this is the
Profe,
structure.
perdón que me interrumpa. ¿Se supone que está mostrando algo? Aparte del video en YouTube.
No escucho, perdón.
Que sí, se supone que está mostrando algo además del video de YouTube. Es que, bueno, no sé si está mostrando otra cosa.
Perdón. Ok, eso es el problema. Gracias que usted dijo. Ahorita mejor que yo puedo compartir mi todo screen. Eso no hay ningún problema de este tipo. So ahorita usted puede ver que aquí hay una... so ahorita, ok, yo explico otra vez. Fura 2, Fura 2 is interacting with calcium and different concentration of calcium will have different excitation intensities. You can see here, highest concentration has got highest intensity of light. This way your excitation intensity can be determined. All right. And then you can use this like these dyes for staining the cells. And then as you are staining the cells, this is the confocal microscopy of the cells. Then what happens here is the internal charge transfer. What happens here is that there is there is a molecule, and this molecule will be interacting with there is a receptor, and this receptor interacts with the ligand. And as soon as there is ligand receptor interaction happening, then there will be conformational change. And as the conformational change happens, then accordingly, this guy will be fluorescent. So like that, you can identify how the fluorescence is introduced. All right. So now there is a first kind of sensor, which is known as intrinsic sensor, chemosensor. What is happening? That means the sensor intrinsically, it will be producing fluorescence. And as soon as you put any other ligand, there is a quenching of the fluorescence. So here, this is fluorescence, and this is quenching of the fluorescence. When there is a molecule which sits onto it, so it doesn't allow to fluorescence. It doesn't allow the molecule to fluoresce like this. So they will stop fluorescence. So this is the way how fluorescence quenching works. Okay. Then that was talking about intrinsic one. Then is conjugate, wherein you have a receptor which is associated. It is not the active site, but it is associated with a highly fluorescent molecule, which is fluorescent in nature. And once it interacts with this whole complex, interacts with this receptor, then what will happen is they will be, I mean to say, once this particular complex is interacting, then after that the fluorescence will stop. There is quenching of the fluorescence. Then there is autoensemblato. That means there is a so autoensemblato means there is a interaction between I mean to say there is interaction between the receptor and ligand, and this time the ligand is fluorescent molecule. And as soon as it is interacting with another non-fluorescent molecule, then this guy will be kicked out, and then now this guy has to create. I'm sorry. Then after that, this complex, what this ball does is they will kick out the previous ball, and then they will occupy that ball. And in that process, you will understand that there is like competition between two molecules to sit onto the same active site. This is idea. Okay. Do you have any doubt, friends? Hello, anybody is having any doubt?
Creo que por el momento no.
Okay.
Gracias.
So ahorita, oh, okay. Ahorita is photo-induced electron transfer. What is happening in photo-induced electron transfer? That this is the complete complex having two active sites. One active site is this, and the other active site is this. And this is the complex molecule. Complex molecule, as soon as it sits onto the receptor, then the whole molecule moves, like there is a conformational change in the molecule itself. And now there is, as there is a conformational change, then there is a fluorescence in the molecule. So this is what is happening, how the fluorescence takes place. So conjugate chemosensors. So what is happening is here, what are active substrates? That means active substrates are, for example, when copper is not sitting. Okay, when copper is not sitting, it becomes an active sensor. When copper is sitting, I mean to say, then there is a change in the fluorescence. Okay, like that. So that is the idea. Okay. Oh God. Okay. So this is the second strategy, conjugate, but I'm not interested in that. I'll just check it once up. Okay. So here again, there are like active substrates. So copper is not sitting, copper sits after that and it forms a proper complex. Then this is a pH sensing system wherein eye of fluorescence. So what happens is TMCA, ATMCA, these are the different molecules which are generated. And then there is like excited fluorophore and bound receptor fluorophore. So it's so then here we have a complete how to say how the molecule interacts with changes the pH, and as the pH changes, the molecule also there is a change in the pH in the molecule itself, and then your stability might be affected like that. So here we have copper, here we have potassium, here you have zinc ions. Okay. Here, so there you can see here, there is this copper. So initially you will find that this whole complex, okay, ATMCA, this whole complex is highly fluorescent. And when copper interacts with it, copper will sit into between NH3, NH2 and NH. So it forms a complex with it and it forms Cu plus 2, copper. And once the copper is formed, then after that, once the copper is interacting with the complex, then after that, this complex interacting with it will have higher, so now then there will be quenching of the fluorescence. In the last thing, so, okay, oh, sorry. This is gone up. Here, this is what is known as, okay, so this was, so this is Zn plus 2, Zn plus 2, Zn plus 2. So zinc ions here, all of them are detecting zinc using the fluorescent markers, all right? So we can have one more detection thing. Wait a minute. Give me a second. Okay, so now I'm showing you zinc sensing receptor. Because zinc sensing receptor is very important for GPR39, that's a protein, or even zinc finger motif. Okay, I'll show you first of all this complex. Where is that? Here. This guy wants to say.
You're probably getting PDFs all the time, right? A teacher might assign you a bunch of PDFs that you have to read.
Okay.
The G protein-coupled receptor 39, or GPR39, is a zinc sensor and is expressed on the surface of intestinal epithelial cells as well as other cell types. Binding of zinc to this receptor activates its G protein, which increases the activities of mitogen-activated protein kinase and protein kinase B, or AKT. The GPR39 undergoes a conformational change when zinc binds, resulting in the activation of the G protein. GTP, bound to the activated G protein, switches with the GDP, and the G alpha subunit dissociates from the other subunits to activate phospholipase C. Phospholipase C then cleaves phosphatidyl 4,5 bisphosphate, producing inositol triphosphate and diacylglycerol. Diacylglycerol remains bound to the membrane, and inositol triphosphate is released into the cytosol and stimulates calcium efflux from intracellular stores such as the endoplasmic reticulum. The calcium activates the mitogen-activated protein kinase and protein kinase B signaling pathways, resulting in 1. Increased synthesis of zona occludens 1 and occluding proteins that comprise tight junctions. 2. Increased synthesis of the sodium proton exchanger that regulates the acid-base balance within the cytosol of intestinal cells. And 3. Increased surface expression of the potassium 2-chloride transporter.
It helps in potassium.
The intestinal cells are then protected from acidic short-chain fatty acids synthesized by the microbiome. They exhibit increased sodium absorption and reduced water loss during diarrhea, as well as increased
I'm just give me a second. Actually, where is the structure? Yeah, it's here. So here we have again, like this is the prototype which has been made for the measurement of hemoglobin, pH, oxygen, carbon dioxide from blood. And all these you can imagine that you can add a receptor, like you can add a fluorescent marker, and then you can identify whether CO2 is existing or oxygen is existing, oxygen hemoglobin is existing or not. So this is the way people have like make the whole idea this kind of polymer oxygen is there, polymeric oxygen is there, and polymeric oxygen is like all of them will be interacting. All the three can interact with the ion, which is very critical. And once they interact, then there will be fluorescence. So like that. Okay, now here what happens is there is a molecule like calixarin. Calixarin is a very complex structure and it changes its conformation continuously depending on the ions. So here you can see here The calixarin is this structure. The first thing, if you want to develop a sensing system, the first thing is you need to be the first thing what you have to do is you have to allow this interaction to happen. Like I mean to say, there is a here there is a molecule, okay, which is interacting with the calixarin, and as the so it will break apart. There will be a conformational change. All right, so this is the ion that is sodium ion. It will sit into this particular zone, will sit into this zone, and then there will be conformational change, and accordingly, you can identify the peak like that. Similarly,
Profe, lo
sorry,
siento, pero ¿qué es el calixare o eso? Es que no entendí esa parte.
calixarin, sí.
Ajá, ok.
¿Otra vez? ¿Yo explico otra vez?
Sí, por favor.
Okay. See, so there is a calixarin molecule. Now, this calixarin molecule is, it will show excimer emission in one condition when all these anthracene, the whole complex is integrated. It will show excimer emission. So it will show emission at a particular wavelength, okay, and some wavelength. So that I will consider that as excimer emission. And so excimer means it is conjugate. So there are two structures which come together, and that is known as excimer emission. And now, if at all this sodium sits into it, then these two structures open. And once it opens up, it's known as monomer emission. That means there are monomer formation, and then there will be emission of light. So in this way, monomer and excimer emissions happen. All right, then there is like planarization. So again, calcium sits into it, and then there is I mean to say calcium interacts with the molecule, with the cells. I mean to say the complex. Again, like calcium and calmodulin, in the same fashion, calcium will interact with any of the dye, and then it will show some fluorescence. So here also, this is known as FRET, fluorescence resonance energy transfer. So what happens here is there are two different colored. There are two differently colored. You can see here, this is one color, all right, of the fluorescent molecule, and this is another fluorescent molecule. So individually, you can see here they're showing different fluorescence. So basically, there is no much fluorescence. All right, or if there is fluorescence, yes, individually they will fluoresce. Now you have added calcium. Now the whole thing changes. I mean to say, when calcium is added, so you can have a complete picture, like you can have the complete structure having FAM and TAMRA. So what will happen is this FAM, which is connected to TAMRA, it will connect. It will give out some energy, fluorescence energy, and then this guy fluoresces. Otherwise, this guy would have not fluoresced at all. Okay, this is self-assembled ATMA for zinc. And this is again for zinc. Chemosensing ensemble. So here what happens is, you can see here, there is a competition between the fluorescent molecule and your ligand, the desired ligand. So there will be like replacement, and when it is bound to the complex, it will not fluoresce. There will be quenching. But as soon as it removes, it gets out from this fluorescence, this guy comes out, and then there is, chemo sensing ensemble, which is happening here. So here you can see that there is a CuNH linkage, and the complex is formed. Okay, so you can see here there are different amino acids, and different amino acids will interact with different molecules, and as they like fluorescent molecules, and as they interact with fluorescent molecule, they will give you fluorescence. So individual amino acid has got affinity towards binding different fluorescent molecules. It's like that. And there is also a concept of there are different plots which have been formed, and these plots are also having some centers wherein there will be they will be detected, and that too which kind of amino acid it is, like it is a dextrorotatory or levorotatory. So that also can be detected using this kind of complex studies. Then self-organized chemosensors, that is quantum dots. So you can see here that these quantum dots, they automatically synthesize and they automatically give the color. Of course, the temperature must be 320, 350 degrees Celsius. But at that particular temperature, you have to just remove the molecule from the solution. And then after that, you'll have different colors. So these are the examples of different colors which we have found. Then after that, this self-organized chemosensors, that is quantum dots. Quantum dots are playing an important role because these quantum dots, they will be interacting with the molecules, with the structures, and then they will be helpful in sensing certain molecules, certain structures, wherever they have got very high affinity. And these quantum dots, you can interact with, like I mean to say, the quantum dots have been used for interacting with certain structures, and once that structures are interacting with each other, then you have a proper sensing mechanism using PET modulation. All right. Then there is again the same on and off complex. So there is a quantum dot. Quantum dot is interacting with some molecule, and then there is a FRET analysis. Due to FRET, you can immediately remove the other structures. Yeah. I hope you are understanding whatever I'm explaining, guys.
Yes.
Okay. I'm just checking. Okay. So I would like to show you how these, because silica nanoparticles are considered to be very, very effective structures for fluorescence. So I will share with you one more diagram, one more video. This I go to fluorescence based sensors. Okay. And how much time? They have not mentioned anything. Okay. And you can see here. Okay. Minute. Okay. Now you can see here one video I'm sharing with you guys. Here. You can see here there are fluorescent molecules. pH 7, and as the pH changes, the fluorescence changes. So as the pH is changing, the fluorescence of the base also changes. Okay. Okay, fine. And there is one more SiO2 based. Okay. Just give me a second and I will show you silica based. Oh God. Nanotechnology. Silica fluorescence animation. Okay. Just give me a second. I'm having this structure. Wait. SiO2 in low pressure. SiO2 simulation. Okay. And there was one video of SiO2 photoluminescence. Animation. It's photolithography, but stepper exposure, positive mask, negative PR and negative mask. Etching of the photoresist. Etching of SiO2. Hashing. Metal deposition of exposed silicon. This is a way. But then I would like to show you something else. Silicon fluorescence. Can you see the video, guys? Am I right? You can see the videos?
Yes, we can.
And you can hear also, right?
Yeah. But it was music, no?
Yes, it was just a music, nothing else. Yeah. And now I'm showing you fluorescence. Sorry, I'll show you that also. But where is this silicon? There is silicon. I don't know.
Así que quieres aprender inglés. Aquí tienes 3 mitos que son completamente falsos. 1. Debes hablar mucho.
Hello and welcome to Nanotechnology channel. I'm Mars.
There is nothing here
Enzyme
explained. Just give me a
nanoparticles
second.
act as
Oh my God. The searching, when I lose it, it takes a little time. Oh. My God, there was a beautiful soul gel method. Silica fluorescence animation. Fine, I'll just skip this because I'm not finding it. Not to waste much time in it. I think this one. Maybe this one, let's see. Yeah, silica
NUVEC is a silica nanoparticle coated with a linker and has a unique structure that traps and protects DNA or RNA within its spikes. The DNA is protected from exposure to nuclease on its journey to antigen-presenting cells. The NUVEC particle attaches to the cell and is endocytosed. Once inside, the linker ruptures the endosome to release the particle and DNA into the cytoplasm, where it makes its way to be transcribed. RNA then translates into the target protein, which is broken into peptides. These peptides are displayed on the surface of the cell and presented to antigen-specific T cells. The adjuvant nature of NUVEC means T cells are strongly activated so that they are ready to attack cancer or virus-infected cells.
Okay, I'll show it again. I'll explain you this, okay?
NUVEC is a silica nanoparticle coated with a linker and has a unique structure that traps and protects DNA or RNA within its spikes.
So these are silica nanoparticles.
The DNA is protected from exposure to nuclease on its journey to antigen-presenting cells.
And then they will throw apart.
The NUVEC particle attaches to the cell and is endocytosed.
And now it's getting inside the cell.
Once inside, the linker ruptures the endosome to release the particle and DNA into the cytoplasm,
all right,
where it makes its way to be transcribed.
DNA gets into it, and then
RNA then translates into the target protein, which is broken into peptides.
Yeah.
These peptides are displayed on the surface of the cell and presented to antigen-specific T cells. The adjuvant nature of NUVEC means T cells are strongly activated so that they are ready to attack cancer or...
So try to understand, this is the silica nanoparticles, and they are all coated with DNA and there will be enwrapped by a polymer. All right, then it moves.
NUVEC particle linker and extracts DNA or spikes. DNA is protect exposure journey to NUVEC particle attaches to the cell and is endocytosed.
As soon as it moves inside the cell,
once inside, the linker ruptures the endosome to release the particle and DNA into the cytoplasm,
and the DNA is released, and DNA is basically responsible to enter inside
where it makes its way to be transcribed.
Do this transcription process.
RNA then translates into the target protein, which is broken into peptides.
Now this particular protein, what is the role of this particular protein?
These peptides are displayed on the surface of the cell and presented to antigen-specific T cells. The adjuvant nature of NUVEC means T cells are strongly activated so that they are ready to attack cancer or virus-infected cells.
Denatured tumor cells. So that's the idea. So with this, you are using silica nanoparticles which are fluorescent in nature, and they will be helpful in killing the cancer cells. It's like that. So they are fluorescence. Here fluorescence plays an important role. And then I'm talking about, they will be responsible for excitation and emission. Of course, or it's the same as other emission fluorescent molecules are doing it. And then there is like then there are many molecules which can attack or attach lead, Pv+2. Now they will interact with Pv+2. Once the interaction happens, the Pv+2 is there, and then it forms a proper Pv+2 interaction happens, and then these molecules will sit onto the surface like this, and as soon as there is like lead sitting onto the surface, what will happen is, so here there is a silica nanoparticles. Silica nanoparticle will be having a long chain like this, and then after that, once this silica nanoparticle is sitting like this, there is a complex formation happening, and then there is like lead which sits onto it. And as soon as this lead is sitting onto it, then there will be emission of some fluorescence. So that is kind of work which people have done using silica nanoparticles.
Profe, de nuevo no está compartiendo. Se quedó en el video. No sé.
Ah, otra vez, pero yo puso ahorita todo de mi. I was sharing the complete screen. Okay, again. So you can see here.
Yes, no.
So yeah, this is the thing. So there is a quartz, then silica nanoparticles, and then they are having a linker attached to it. And then when the molecule is sitting onto it, then the molecule will, like an analyte, will sit onto it. And as the analyte sits onto it, there will be, like, they will emit some fluorescence. So this is the idea behind using these the silica nanoparticles. So that's the idea. All right. Then there are many structures like this, pebbles. Okay, I don't think so. This is very critical. And this is the way your detection of Pb+2 happens, as we have already discussed how silica nanoparticles are useful for detection of lead. I think we have almost completed these sessions. Okay, nano HPLC. And how zinc detection happens, and why zinc detection is important. So this is, I will show you. Okay, yeah, it's fine. I don't think so. It's fine. I think it's like perfectly done. There's no need to. Like, these are repeating so many examples of zinc with the fluorescent dye is being shown. That's the only thing. And this is silica core, which we have already discussed. With this, we have finished the session. And I would like to show you one more last. I'll stop sharing it. And I want to show you one more very important video. Wait a minute. All right. Okay. So here I'm just sharing with you. I'm sharing with you something with respect to microfluidic chips. So how these are being made, etc., etc. So all those things will be shown here. I'm just sharing it with you guys. Yeah.
Tú no paras. Rexona tampoco.
Our S1 chip recreates the microenvironment of an organ using living human cells. It can be used to emulate a number of different organs and disease states, providing a real-time window into the inner workings of human biology. The chip features two fluidic channels that form the necessary architecture for building tissue-to-tissue interfaces. It's made from a flexible polymer that's both stretchable and durable.
VDMS.
Its transparency allows researchers to observe real-time cellular behavior inside the chip's fluidic channels. The top channel is 1 millimeter wide by 1 millimeter tall. The bottom channel is 1 millimeter wide by 0.2 of a millimeter tall. The channels are separated by a thin, porous, stretchable membrane that allows for cellular communication between the two channels. It's coated with extracellular matrix proteins, promoting cell attachment and assembly. Different cell types can be seeded into the top and bottom channels. Mechanical forces can be applied to the chip, allowing cells to experience forces similar to breathing or peristalsis. These can be critical determinants of cellular function, cell
So here you
signaling,
can,
and gene expression.
so you can see here, there are, oh God, where is the video? So you can see here that there are like two different kinds of cells we can grow on the top as well as at the bottom. Okay. And they are all separated by a very good membrane, a flexible membrane.
Breathing or peristalsis. These can be critical determinants of cellular function, cell signaling, and gene expression. Both channels can be configured to flow media, blood, or air, and the biochemical environment the cells experience can be precisely controlled by providing nutrients and washing away waste. Immune cells can also be introduced into the chip, recreating mechanisms involved in the body's immune response. Our S1 chip is designed to help us predict how medicines, chemicals, and foods affect human health. It's one part of the human emulation system, which is allowing us to gain a deeper understanding of human biology.
This is how a microfluidic system is working, and organ on a chip is being developed, and there is oh God, wait a minute, I'm just sharing with you one more. Yes, I think this one, I hope this one works well for you. Yeah, I think this one is, this is a step-by-step formation.
During an experiment, pause the program and remove.
All right, this is a step-by-step formation of the chip.
Standard cell culture
can you
techniques
see
fail to
the
provide
video?
insights into the complex, multifaceted interactions that take place in a living
I'll again share it, otherwise, maybe. I'm just sharing it to you with you again.
Sí, sí se ve el video. So, yo no.
Otra vez, ¿no? It's a 5-minute guide, all right. ¿Puede ver ahorita?
Sí, sí se ve.
multi-organ system. Organ-on-chip devices allow your team to run studies on living human cells and tissues. CN Bio's Physiomimics organ-on-chip device, also known as PMX-OOC, allows you to model human biology in the lab by creating human-relevant 3D tissue models. PMX-OOC is a compact and easy-to-use microphysiological system that can be used to culture system.
Yeah.
Standard cell culture techniques fail to provide insights into the complex, multifaceted interactions that take place in a living multi-organ system. Organ-on-chip devices allow your team to run studies on living human cells and tissues. CN Bio's Physiomimics organ-on-chip device, also known as PMX-OOC, allows you to model human biology in the lab by creating human-relevant 3D tissue models. PMX-OOC is a compact and easy-to-use microphysiological system that can be used to culture healthy or diseased tissue models, study multi-organ interactions, and investigate immune cell function. Step 1. Simple preparation. To get started with the PMX-OOC, simply dock the MPS plate of choice onto the MPS driver. Once docked, prime the MPS plate with cell culture medium. All MPS plates contain microfluidics, which control medium flow within individual cell culture wells. Using the liver MPS plate, different cell types of the liver can be seeded into 3D scaffolds to form tissue structures that mimic the function of the human liver. The multi-MPS T12 plate is used to model barriers, including those found in the gut, lungs, and kidneys. Simply seed cells of choice onto the T12 inserts and transwells and transfer these into the T12 plate. Step 2. Long-term experiments, minimal user input. The PMX-OOC is compatible with standard cell culture incubators. Transfer the MPS driver and docked MPS plate from the biological safety cabinet to a cell culture incubator and secure the MPS onto the docking station. The intuitive software of the PMX-OOC controller automates control over the microfluidics, enabling long-term experiments with minimal user input. The microfluidics perfuse cell culture medium through or across tissue structures, providing oxygen and nutrient diffusion, as well as biomechanical stimuli. Step 3. Flexible experimental design. The PMX-OOC open well design allows easy access to cells and media for dosing, sampling, and analysis. During each experiment, cells can be dosed with small molecule compounds, antibodies, hormones, or genetic engineering reagents. To do so, remove the MPS driver and MPS plate from the incubator. Dose the MPS plate with the compound or reagents of interest. Then, return the MPS driver and plate to the incubator. Step 4. Sampling and analysis. Media and cellular endpoint analysis can be easily performed from PMX-OOC samples. During an experiment, pause the program and remove the MPS driver from its docking station. Media can be sampled into a 96-well analysis plate, for example, to test for the presence of soluble biomarkers. To analyze the cells from the experiment, remove the scaffold from the MPS plate for imaging or for lysis of the tissue. Scaffolds from MPS plates can be imaged by many approaches, including confocal, EM, and histological staining. Performing omics analysis on PMX-OOC samples will enable detailed understanding of...
What you can do is, once the cells are being grown inside the microfluidic or organ on the chip or organoids are formed, then you can simply find out using this particular technique whether the cells are there, living, dead, and what are the structures, what are the morphologies. Everything you can understand using this technology, using this idea.
Human cells' response to a particular drug or chemical. Step 5. Multi-organ experiments. The PMX-OOC device allows for the performance of either single or multi-organ experiments. PMX-OOC MPS plates are compatible with commercial inserts, tissue-specific scaffolds, and spheroidal cultures for easy onboarding of validated or bespoke cell models. Use the PhysioMimic software to automate your microfluidics. Enjoy precise control over inter- and intra-organ flow rates. Enable precise control over cell culture conditions and on-platform pharmacokinetics, all at the push of a button. Step 6. Create advanced human cell cultures and organ mimetics. PMX-OOC is a streamlined, low-footprint lab instrument, compatible with a variety of PMX-OOC MPS plates to enable the study of various human tissues and disease processes. PMX-OOC quickly increases your in-lab capabilities to more accurately recreate human-relevant cell culture models. Whether you want to maximize the potential of your existing cultures or undertake complex multi-organ studies, PhysioMimic's OOC is the cutting-edge cell culture solution for your research.
Perfect. So I hope you have understood. I'm talking about which particular system. Can I see all your faces if it's possible? Because this is the last session and I would like to have a good one-on-one. Is it possible for you guys? If it's possible, it will be great. And I think we can have a photo together.
Yes, one moment.
Thank you. Thank you. And anybody can take photo here? Or else I have to take photo with this maybe. But I don't know whether... I think one more thing, I can have this thing, right? Imprimir pantalla kind of. Yeah, I will do that. Or else it's like recording is going on. So remove the... So this was the complete idea which I could give you. using this. I hope with this particular knowledge, I think there will be a great understanding about the subject, and you could understand well how these cells, how the cells are behaving, how biosensors are being developed using different techniques like using optics, using genes, genetics, DNA, and using light and fluorescence basically. So these are the different things. And what is the motive behind doing this? The motive is only one, that inside the cancer, how to detect cancer, how to detect different pathogens. Pathogenesis can be detected using these techniques. So this is what I hope. I think I have given a complete overview of what is happening inside the cell, and outside the cell, how the detection happens. So thanks a lot, guys. I hope I have helped you, or maybe something has happened, something I could help you in getting instigated or stimulated using this subject. So please, if you can give me little bit of your feedback, little bit, so that I can understand that what I should not do in future for my future students, and what I should do for my future students. So please tell me whatever, like, please give me your feedback. Raimundo, I think you can start.
Yeah,
Is it possible, Raimundo?
yeah, sorry, I am too busy.
Sorry. Um, Hello.
I am active now. Hello?
Yes, I can hear you. I can hear you. Yeah.
Yeah, I really like organometric systems, but never in the university
Your
with
voice
our
is
tag.
there is an echo coming. Please, if you can, there's an echo coming, echo. Maybe by the time Raimundo is explaining something, before that, like we can talk to maybe Miriam Cepeda.
Yes.
Yeah, Miriam, tell me.
¿Le molesta si le digo en español? Es que realmente sí me
Está bien,
costó.
está bien, no hay problema, nada, nada.
Gracias. Pues la verdad se me hizo muy interesante. Creo que algunos temas ya los hemos visto a lo mejor y superficialmente en las otras módulos, entonces creo que fue bastante bueno el que pues, profundizáramos un poco más en el tema. Honestamente creo que me gustó mucho el artículo y creo que sí me descargué el del otro equipo para leerlo con más calma. Y se me hizo como una buena actividad el que pues nos juntemos y aunque obviamente no pudimos leerlo todos, todo el artículo,
Sí.
estuvo muy bien que pudiéramos compartir lo que se nos hizo más importante y todo eso. Y creo que estuvo bastante... Ay, bueno, creo que algunas definiciones sí me ayudó mucho que las volviera a explicar y que tuviera esa paciencia en que hay algunas cosas que a lo mejor y a mí como persona no se me pegan tan fácilmente, entonces creo que estuvo bien que nos lo explicara de nuevo. Y pues sí, creo que me llevó muchas cosas. Muchas gracias.
Gracias a usted, Miriam. Muchas gracias, muchas gracias. Sí, Dennis Ramos. Yeah.
Yeah, I think it was a really interesting topic. It's something that I think is really the future of my career, that is paramedic. So it was really important to me to learn about the lab on a chip, all of the different sensors that will look at this. So I really appreciate all the knowledge that you give us.
But then could you like I'm asking you that whether you could get benefit out of this, or and if you feel that no, I think this should have been better in this sense or that sense, that feedback is also acceptable. There is no problem. But you can tell me. You can tell me.
Yeah, I think it was really good. Just I will do it a little bit more. How do you say it? Con más participación de los alumnos, un poco más. Olvidé la palabra.
See, yeah. The students needs to participate, but how? Tell me that's my question, because I tried yesterday using this articles and all. So I was thinking that maybe the students are coming together and they're discussing about the article, and then they have to, they need to explain well their concepts, and that was one aspect. But I really, I'm not understanding how these students need to come and have a group discussion. That is something which I need to understand.
Yeah, it could start with that, and other or of teachers is do something like that, put us in little groups, give us something to discuss, and then we come back to the session and put it the things that we understand or the things that we find in one table.
Yeah.
So that's the form. I think it's a really good form, and it's easier to the students to put all the knowledge in one place and to the teacher to see how much they understand about the topic.
Perfect. That's really great. I'll incorporate that idea in the next sessions. Yeah. Thank you, Dennis. Thank you. And if at all you guys want to come for the yeah, Dennis, nobody should go because we have to take a photo. All right, please, just I want to get a feedback first. Answers, yes.
Yeah, well, for me, it was a great course. I learned a lot and I reviewed some topics. And I hope to be able to use it in a project in the future. And well, the class, the dynamic, it was for me good. I don't have problems. I like that the teacher speak a lot because, ay, no me acuerdo cómo se dice, si soy auditiva en las partes de cómo aprendo. Entonces,
sí.
So for me, it was good. Yeah, thank you.
Thank you, Aranza. Muchas gracias. Sí, Jose Valentín Arroyo Martinez. Yeah.
Yeah, well, as well, I also enjoyed this model. It was so interesting to take it in English because the other ones are in Spanish. I would say that the dynamic of class was good, but also maybe adding some more activities like the one that we did yesterday. Maybe one more or two more. It's a good strategy to learn by ourselves. So I will say that.
So you liked yesterday's idea that we did with making breakout rooms and all. You liked that idea?
Yeah, that's a good, as I said, I think it's a good way to develop our self-understanding of some topics.
Very good. Because I was I was worried about that idea because we just spent one whole lecture on a topic, and just this was like interaction, like it went on only with interaction, and sometimes sometimes we feel like the professors feel that, hey, I've not taught the topic. So sometimes this kind of, I was little worried that the students must have been feeling something wrong, that, hey, the professor has not done anything in this. He has just made us to work and he has not done anything. So I was like under that impression that like how the students are feeling. But as you're saying that, okay, you are positive about it, then I felt I am feeling great because I want the students to understand the concepts. The student need to develop. That's the idea. But thanks, Jose Valentin, because I was I've got a positive picture now. Thank you for this. Thank you. And Rodrigo Solis, por favor. ¿Qué es esta? Trains, cars, departure. Okay. Yeah.
Can you hear me?
Yes, yes, I can hear you perfectly.
So, oh, well, I want to say that I enjoy this course. I enjoy like the way you like gave it to us with the articles, going into a group. I really enjoy that. And well, this type of subjects, I really like them because, well, I'm more in the biomedical field, but this type of subjects, we don't really get in touch of them. They show us more stuff like, I don't know, machines and stuff like that. So going on this side, actually, a bit of a game changer for us. So, like I said, I really enjoyed it. It opened my mind to like not going join, not just going to a hospital, maybe a research lab or other stuff. So, for my behalf, I want to thank you for opening up my eyes and showing me that I can go into another place, another field like that.
Very good, very good. Thank you, Rodrigo. Thank you. Well explained. Yeah. Isaias Flores.
Yep, can you hear me?
Yes, I can hear you.